Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is meant for 7th October 2020, and we are into the revision mode. As we completed revision of chapter 1, we move ahead with the chapter 2, biological classification. And what we do is we also do, along with the revision, the NCRT question answers that I will be posting in your group, WhatsApp group. So you need to keep one 200-page notebook. So first write the revision notes. After writing revision notes, answer question answers of the NCRT, and then on alternative days or once in 3 days I will be posting MCQs of that particular chapter, and then reasoning and assertions questions. So with that you will have a firm understanding of the concept, and with that you have a lot of confidence to face the examinations. Along with this, as and when we complete one revision notes, related experiment also I will be dealing with that. So you need to maintain one biology record, and that record is having a value of 6 marks. So you need to maintain a record, and then at the end of the experiment there will be viva questions. Normally external examiner who comes will be asking you that questions related to that experiment, what you have done, what is your observation, what is the result of it, what are the precautions you have taken. So based on that, lot of viva questions will be there. So in that also I will train you. So keeping all this in our mind, you need to maintain two things. One 200-page classwork, or you call it homework. Of course, classwork now, this itself, videos itself will become a classwork to you. But you need to maintain one homework. In that homework, what I am expecting is revision notes. Whatever revision notes I am posting it every day. Second, NCRT question answers, your textbook questions. And the third one is the MCQs that I will be posting. Fourth one is assertion and reasoning. And fifth one, that you need to keep separate biology record. And in that record, you have to write the experiments that I am going to tell you. Plus, there will be virtual labs. I will be posting that circular in your group. So you need to log into that particular website, and it will be something like online practicals. You doing online practicals. Okay. Anyhow, when you come back to the class, physically you will be doing it. Until then, just to get familiarized with various experiments that you need to perform, I will be posting that website also. So you can log in and see the experiments that you perform, right? Moving ahead, what is biological classification? In first chapter only, we were talking about that classification, taxonomy, all that. So it is the scientific procedure of arranging organisms into groups. Remember, taxonomical hierarchy and subgroups on the basis of similarities and dissimilarities. This is the thing. And remember, the similarities, dissimilarities, we related this with the key and lead. So keys and lead will help us to make the similarities and dissimilarities. And based on that, we place the animals and plants into different groups in various hierarchy categories. So what is the importance of classification? Why should we do this biological classification? It is not possible to study every organism. Study of one or two organisms of a group gives sufficient information about essential features of the group. At least, see, phylum, order, class, this much will have general characteristics. Like if you study cockroach, you have almost, say, some very common interesting characters of phylum Arthropoda, like that. So when you study a flowering plant, you know the division angiosperms. So some of the important characteristics you will come to know. And it helps in identification of new organ. Based on the characters that you observed in a particular animal, we can relate that and we can find out the new organism. Classification helps in knowing the relationship among different groups of organisms. So we combine different classes, different orders, and come up to the division or phylum and study what is the relationship between the various classes and orders under the phylum. And we can have interphylum comparison also we can have. So the organism of the past cannot be studied without a proper system of classification. So we need proper classification to study an organism that was existing in the past. It is no more. So there are different varieties of classifications. Artificial system of classification, natural system of classification, phylogenetic system of classification. So what is this artificial system of classification? Only one or two morphological characters for grouping of organism is used. So here we are talking only external characters. Morphology is study of external characters. So flowering and non-flowering plants, enema, enema, Aristotle classification. Aristotle used this. But just based on morphology, we cannot make all the organisms, because anatomy is there, biochemistry is there, so those things will crop in. So hence we don't depend much on this artificial system of classification. So what is natural system of classification? It takes into consideration comparable study of number of characters so as to bring out natural similarities and dissimilarities, and hence natural relationship among organisms. Who is to be created with this? Bentham and Hooker. And then phylogenetic, phylogeny, evolutionary relation. Based on the evolutionary relationship of organisms, this phylogenetic system is arranged. In this system, organisms are classified on the basis of their evolution on Earth, from primitive to highly evolved. From primitive organisms, we go up to the most evolved organisms, and Engler and Pruntil classification, and Hutchinson. This is very famous way of doing it. And then depending upon the type of system of classification, organisms are classified into following kingdom system. Most accept one kingdom system of classification. We got two kingdom. All the plants that are available on the earth come under kingdom Plantae, and so as all the animals that are available that live on land are Animalia. So basically two kingdom classification became PLAP because there are viruses, for example. They don't come under, say, living or non-living. Similarly, there are some organisms which have both plants characters, animal characters. And what about bacteria? So lot of questions were there. So two kingdom classification became a PLAP. And then three kingdom, Plantae, Protista, Animalia. So the Protista belongs to the primitive organisms. But here also conveniently fungi is forgotten. So this three kingdom also became PLAP. And most accepted one is the five kingdom, Monera, Protista, and fungi has come, Plantae, Animalia. So two kingdom system of classification organisms are grouped on the basis of presence and absence of cell wall, as proposed by Linnaeus, father of taxonomy. Your UPSC question, father of taxonomy, Linnaeus. Three kingdom, Haeckel, proposed by Haeckel, separated unicellular animals, algae and fungi on the basis of lack of tissue differentiation, and the new kingdom Protista was introduced. What about five kingdom? Very famous, Whittaker. R.H. Whittaker proposed this. Whittaker divided all organisms into five kingdoms in order to develop phylogenetic classification, and that is most accepted one as on today. So first one is in the five kingdom, Monera. The kingdom includes all prokaryotes. What do you mean by prokaryotes? Pro means primitive. Karyotes having carrion nucleus, primitive nucleus. Instead of having a nucleus, they will be having a small nucleus, conspicuous one, nucleoid we say. Nucleoid, it is not nucleus, nucleoid. So mycoplasma, bacteria, actinomycetes and cyanobacteria, all these come under Monera. And what are the basic characteristics of kingdom Monera? Kingdom Monera, all are unicellular, okay, one cell body, prokaryotes, the nucleus is primitive nucleoid and contain most primitive living forms. The cells are microscopic, we need microscope, we can't see them with naked eye. Cell wall is generally present. Genetic materials are not organized into nucleus and contain naked DNA. Membrane bound organelles are absent, so organelles also not clear. Reproduction is asexual except gene recombination. Flagella may be present and are of single strand. This will help in locomotion. Example, blue-green algae and bacteria. And bacteria are the most abundant microorganism that can survive in all kinds of climate. So you got based on the shape, bacteria is of 4 types. One is first coccus, spherical shape, round one, spirillum, coiled shape, and rod-shaped bacillus, and comma-shaped vibrio. So bacteria is of 4 types. And moving ahead, this is that cocci, spherical, bacillus, spirilla, that is spring-like, and comma-shaped vibrio. They are group of most primitive prokaryotes which live under most hostile, means not conducive conditions, like extreme salty area. So bacteria that grow in salty area are called halophiles, and bacteria that grow in hot springs, they are called thermoacidophiles, and bacteria that grow in marshy areas, they are called methanogens because methane will be there. They differ from other bacteria in having different cell wall structure, absence of peptidoglycan. In cell wall, this will not be there. Methanogens are present in the gut of several ruminant animals like cows and buffalo. So methanogens will be there, which is responsible for production of biogas methane from the dung of these animals. That's why the dung of the animals will be smelling, methane smell will be there. And next, eubacteria. Eu means clear. They are called true bacteria. They contain rigid cell wall. If motile, contain flagellum. Cyanobacteria or blue-green algae are gram-positive photosynthetic bacteria. They contain chlorophyll A and carotenoids. That's why gram-positive photosynthetic we said. They may be unicellular, colonial, or filamentous. Freshwater, marine, terrestrial. That's why we said omnipresent. They are present everywhere. Some of them have specialized heterocyst cells to perform nitrogen fixation. In that heterocyst, there will be special structure and they are responsible for nitrogen fixation. Example, nostoc and anabina. And chemosynthetic bacteria oxidize inorganic substances like nitrate, nitrite, and ammonia to produce energy and help in recycling of nitrogen, phosphorus, sulfur. So that is chemosynthetic. The heterotropic bacteria are most abundant. Hetero means different. They act as decomposers. That's why thanks to them. They are helpful in production of curds, antibiotics, fixing nitrogen in leguminous plants. Some of them are pathogenic, and when they are pathogenic, they cause diseases like cholera, typhoid, tetanus, and citrus canker. So here, many times in your UPSC ND exam, there will be lot of questions on the disease causing bacteria. So you must know the name of the bacteria and what disease it causes and how it is caused. So you must know the carrier also, agent also. Okay. So bacteria, there are 2 types, autotrophic and heterotrophic. Under autotrophic, photosynthetic, blue-green algae, and chemosynthetic, of course, just now we verified that they break the chemicals. And third one, mycoplasma. So they are the simplest free-living prokaryotes. They are also known as, yes, always you will ask you this expansion of PPLO. P for pluro, okay. Another P for pneumonia, of course P is their spelling you see. Like, L for like, organism, O. So PPLO, pluro pneumonia like organism. They lack cell wall. They can survive, this is the important thing, they can survive without oxygen. So many times we will ask you this question. They cause diseases in plants and animals. So name the bacteria that can survive without oxygen is PPLO. And last, Protista. Kingdom Protista includes chrysophytes, dinoflagellates, euglaids, slime mold, and protozoans. So here also Kingdom Protista, I think we will deal with it tomorrow, and general characteristics also we will talk, and we will talk individually about this. And what you must remember in this is dinoflagellates. This is important group. Okay.